Have you ever really stopped to think about what's inside a vial of insulin? Or a dose of one of today's most advanced cancer drugs? Well, today, we're going to pull back the curtain and go inside the microscopic factories that build these incredible, life-saving medicines. It's a world of unbelievable complexity and precision, so let's jump right in.
So that's the big question, isn't it? How do we get from a simple piece of genetic code, basically a digital blueprint, all the way to a physical vial of medicine that's ready for a patient? Well, it's not magic. It's an amazing field called bioprocessing, and we're about to break down exactly how it all works. Here's our game plan. We're going to follow the entire journey. First up, we need the blueprint. Then we have to choose our tiny microscopic factory workers. After that, we'll fire up the production line, run it through some seriously intense quality control, and finally, we'll look at the most important step, making sure every single dose is absolutely safe. All right, let's dive into part one. You know, just like any factory, you can't build anything without a set of instructions, a blueprint. In the world of bioprocessing, our product is a protein, and that instruction manual is written in the language of DNA. So what is the product that's actually coming off this microscopic assembly line? It's something called a therapeutic protein. These things are literally the powerhouses of life. Think of them as the tiny machines doing all the heavy lifting in our bodies, like antibodies that hunt down viruses or hormones like insulin that keep everything in balance. Bioprocessing is all about harnessing the cell's natural ability to make these amazing molecules. This image here shows you how that instruction manual is organized. It's kind of like a set of nested folders. You have the master plan, the DNA, which is coiled up tightly into chromosomes, and within that DNA, you have specific sections called genes. Now here is the absolute key takeaway. One gene is the recipe for one specific protein. That's it. It's an incredibly precise system. Okay, so we know the recipe exists. But how do we get our factory workers, the cells, to actually read our specific recipe and not some other one? Well, that's where the genius of genetic engineering comes in. Scientists can literally cut out the specific gene for, say, insulin and paste it into a host cell. And just like that, you've reprogrammed the cell. It becomes a dedicated factory with just one job, to churn out our target protein over and over again.
All right, we've got our blueprint loaded. Now comes a really critical decision. Who's going to do the actual work? We have to pick our cellular factory for the job. And broadly speaking, it boils down to two main candidates. So take a look at these two. On the left, you've got your prokaryotic cell, something like E. coli. Think of it as a simple, no-frills workshop, small and efficient. But on the right, that's a eukaryotic cell, in this case, a Chinese hamster ovary cell, or CHO cell for short. This thing is way more complex. It's like a sprawling, high-tech manufacturing plant with all kinds of specialized departments. Let's really break down the pros and cons here. You look at prokaryotes like E. coli and wow, that doubling rate is insane. A new generation every 20 minutes. They're also really tough. But, and this is a huge but, they're simple. They can't do complex finishing touches on proteins. Eukaryotic cells, on the other hand, are slower and way more sensitive, but they have a superpower. They can perform what are called post-translational modifications, like adding little sugar chains to the protein. For many of today's most complex drugs, you need those finishing touches for the medicine to work in the human body. So it's a classic trade-off. Do you need raw speed, or do you need sophistication?
Okay, blueprint is ready. Factory has been chosen. It is time to flip the switch and start the production line. The whole goal of this phase is to take a few of our specially engineered cells and get them to multiply into an army of billions, all of them churning out our precious protein.
Now you can't just grab any old cell and hope for the best. Consistency is everything in this business. That's why every single production run, without exception, starts from a cell bank. This is basically a master copy of our perfect, genetically engineered cells, which have been grown up and then carefully frozen in time in liquid nitrogen. This is mission critical for making sure that batch number 1 and batch number 1000 are absolutely identical.
So once we thaw out those cells and give them some food, they start to grow. And they don't just grow randomly. They follow this very predictable pattern you see on the graph here. This isn't just a biology textbook diagram. Understanding this growth curve is the absolute core of managing the whole production process. Let's walk through this journey. First, you have the lag phase. The cells are just kind of getting settled in, adapting to their new home. Then, boom, the log phase hits. This is where they go crazy, dividing exponentially. It's a population explosion.
After a while, things level off in the stationary phase, as food starts running low and space gets tight. And finally, you get the death phase, as waste products build up and become toxic. The real skill, the art of bioprocessing, is knowing the perfect moment to stop everything and harvest, right at that sweet spot at the peak of the stationary phase to get the absolute maximum yield.
And where does this massive cell party happen? It all goes down inside something called a bioreactor. Now, don't just picture a big metal tank. This thing is more like a luxury, all-inclusive resort for cells. The temperature is perfect, the 5-star food is delivered on time, the oxygen level is just right. Every single variable is controlled down to the tiniest detail to keep those cells happy and productive.
Just look at the engineering here. You've got lines feeding in fresh media, that's the cell food. You've got sterile air bubbling through. You see all those probes and sensors? They're constantly checking things like pH and temperature, and a computer is making tiny adjustments 24/7. That thing in the middle, the impeller, it's just a big gentle stirrer, making sure every cell gets its fair share of nutrients. It's a completely self-contained, perfectly managed little world, all built for one purpose: making protein. Okay, so fast forward a few days or even weeks. The bioreactor run is done, the cells have worked their magic, but now we have a huge new problem. Our valuable protein is swimming around in a messy soup with billions of cells, leftover nutrients, and all sorts of other cellular waste. So the next big challenge is, how do we fish our product out of that complex soup?
The process starts with the harvest. We literally drain the entire contents of the bioreactor. This messy mixture is called the broth, and the very first job is a big, crude separation. Think of it like straining pasta. We just want to separate the liquid part, which has our protein in it, from all the solid gunk, the cells themselves. Now, for the really precise, fine-tuned separation, the undisputed champion technology is chromatography. The basic idea is actually pretty simple. Imagine you have a mix of sand and salt, and you pour water over it. The salt dissolves and flows with the water, but the sand stays behind. Chromatography works on a similar principle, but at a molecular level, separating molecules based on properties like their size or charge.
But we can take this idea to a whole new level of precision with something called affinity chromatography. And honestly, this lock and key analogy is the perfect way to think about it. Imagine our target protein is a very specific lock. Well, with this technique, we're going to build a key that fits only that lock and nothing else in the entire mixture.
So here's how that lock and key system works, in 3 steps. First, binding. We pour our messy protein soup through a column that's packed with tiny beads, and attached to every bead is our custom-made key. As the soup flows through, only our target protein, our lock, gets snagged and captured. Everything else, all the impurities, just washes right through. Step 2, washing. We rinse the column with a special buffer to wash away any leftover junk. Now the only thing left inside is our protein, securely locked onto the resin. And finally, step 3, elution. We change the liquid again, and this new buffer basically forces the key to let go of the lock. And out flows our protein, now incredibly pure, ready to be collected.
Okay, we've designed it, we've grown it, we have purified it. We are so close, but we are not done yet. Because now comes what is arguably the single most important challenge of the entire process, making absolutely 100% certain that this product is sterile and safe to go into a human being. In this world, contamination is public enemy number 1. And the scary thing is, it can come from absolutely anywhere. The people working on the line, the raw materials you start with, the pipes in the tanks, even just the air in the room. Every single element is a potential threat that could ruin a multi-million-dollar batch of medicine.
And that leads to this quote, which is basically the golden rule for this entire industry. You see, the strategy isn't about finding and killing bacteria after they get in. It's about designing a process, an impenetrable fortress so perfect that contamination can never, ever get in the first place. The entire philosophy is built on prevention, not reaction.
And that really leaves us with the immense final challenge, doesn't it? We live in a world that is just swarming with invisible threats, bacteria, viruses, molds. So how on earth do you build a manufacturing process so robust, so airtight, that you can guarantee the absolute safety of every single dose? It is a monumental task. And for the patients who depend on these medicines, there is absolutely no room for error.